So now we will see how this is done. Okay. So in this particular topic, we will see metabolism. Now this metabolism particularly defines synthesis as well as degradation, that is anabolic reactions as well as catabolic reactions. Both we have to learn regarding this glycogen. Means how glycogen can be synthesized, that is anabolic reactions, and how glycogen can be degraded, that is a catabolic reaction. Okay. So now first, before learning about glycogen synthesis and glycogen degradation, first we have to know what is glycogen, right? We have to understand what is glycogen, what are the important features of this glycogen, and what are the different bonds that are involved in the glycogen formation. Okay, so glycogen is a storage form of glucose in the animals. So in plants, what is the storage form of glucose in plants? Yes, anyone? What is the storage form of glucose? So similarly, in case of animals, storage form of glucose is known as glycogen. Now, this glycogen is majorly stored in the skeletal muscles and liver. It is majorly stored in the skeletal muscles and livers. Now, the role of the role of this glycogen in skeletal muscle and liver is different. How? In muscles, it helps in synthesis of ATP during muscle contraction. Whenever the muscles are in contraction, these glycogen molecules are utilized and degraded into glucose. And now these glucose molecules undergo series of different catabolic reactions, leading to synthesis of finally ATP is synthesized. So the function of the skeletal muscle is majorly to synthesize ATP, while the role of this liver glycogen is to maintain the concentrations of glucose during fasting. That is very important. Whenever a person is under fasting conditions, at that time glucose is the main source of energy. So whatever the stored glycogen is present in the body, that is utilized for synthesis of glucose. Means now glycogen is being degraded into glucose, and that glucose is utilized, and it is helpful for maintenance of blood glucose levels during the fasting stages. So the role of liver glycogen is to maintain the blood glucose levels during fasting conditions, while the role of muscle glycogen is to provide ATP during muscle contraction. Next, coming to the structure of this glycogen. You can see the right side, there is a picture of a glycogen. The first is the first is a whole structure of glycogen, and the second is a zoomed portion, how these different bonds are formed in the glycogen. Okay, so now this glycogen is a branched chain polysaccharide. Now it is a branch, and you can see these different orange portions. There is a branching occurs, and all the white and orange portions are regarding the glucose molecules. So particularly alpha D glucose is present in this glycogen. Okay, so now these alpha glucose molecules are linked together by either alpha 1,4 linkages or alpha 1,6 linkage. So now what is the difference between these two linkages? If it is alpha 1,4 linkage, then a straight chain glycogen is obtained. You can see these all the white portions you can see, these are all linked by alpha 1,4 linkages. While whenever the branches occurs, you can see this is a branch, now this is a branch. So whenever this branch occurs, the two of the glucose molecules that are involved in the branching, only those molecules will have this alpha 1,6 linkage. Okay, this originally, when you see the zoomed structure, you can see now this is a first position, second, third, fourth, fifth, and CH2OH will be sixth position. Okay, so CH2H is a sixth position. You can see there is a branch formation in this. Orange molecules are branched glucose molecules. So at the first position, hydroxyl group, and the sixth position of the second glucose molecules, that is CH2OH. Now these both are involved in bond formation. And whenever this alpha 1,6 linkage occurs, means you have to understand that there is a branching of this glucose chain. And similarly, you can see in a straight chain, there is a 1,4 linkage. At the first position of one glucose and the fourth position of the second glucose, there is a linkage. Hence, it is known as 1,4 linkage. And these 1,4 linkages are present only in the straight chain molecules, while 1,6 linkages are present during the branches. Okay, so this is the important difference you have to remember. In a glycogen, there are two different bonds. One is alpha 1,4 linkage and alpha 1,6 linkage. That helps in branching of the glucose, these glycogen molecules. Now, these glycogen molecules are stored in the form of granules in the cytosol. Okay, they are stored in the form of granules within the cytosol. Now, cytosol has the majority of the enzymes. Yeah. Anyone, please just move. Who is this? Just check who is. Okay, fine. So now these glycogen, whatever stored in the cytosol in the form of granules, now the cytosol contains majority of the enzymes that are helps in synthesis as well as degradation. Cytosol contains enzymes that are responsible for synthesis as well as degradation of glycogen. Both synthetic as well as degradative enzymes are present in the Akash. Please check your microphone. Akash. Okay, fine. Sorry for the disturbance. Yeah. So now these granules are present in the form of cytosol. So now glycogen synthesis as well as degradation both will take place in the cytosol itself, because whatever the enzymes that are involved are both present in the cytosol only. Hence degradation as well as synthesis. But you have to remember, very important here is, the enzymes whatever are present for synthetic mechanisms are totally different, and whatever the enzymes are responsible for degradative methods are totally different. So here two set of enzymes are present that are responsible for synthesis as well as degradation, both the separate enzyme set of presence. Okay, so this is a very important uh, during this glycogen metabolic pathways. Now coming to pathway of glycogenesis, means synthesis of glycogen. Okay, so how this glycogen can be synthesized? So here for synthesis of glycogen, majorly requirement is ATP and UTP. This is very, very important. UTP is very important in order to synthesize UDP glucose. The very first pathway is synthesis of UDP glucose. So for this UDP glucose synthesis, now UTP is required, that is uridine triphosphate. Okay, so this is adenosine triphosphate. Now this is uridine triphosphate. Now this UTP is utilized for glycogenesis. The different pathways, or you can say different steps that are involved in this glycogenesis is, first is synthesis of UDP glucose from UTP as well as glucose molecules. Second is requirement of a primer to initiate glycogen synthesis. So now this primer, where does this primer comes from? Means already the glycogen whatever is present, when it undergoes degradation, some part of that glycogen is stored, and now that stored part is utilized for again initiation of this glycogen synthesis. Whatever the glycogen is stored, it also gets degraded and also new glycogen is formed. So whatever the degraded products are there, out of that some part of the glycogen is remained as such. It will be not degraded. And that remained part of the glycogen is utilized for synthesis of glycogen itself. So that part that is unmetabolized, that is left unmetabolized, is utilized to initiate the glycogenesis. Until and unless this glycogen part of that or the fragment of that glycogen is not present, then this glycogenesis will not be carried. This is very important in order to initiate glycogenesis. Next is elongation of glycogen chains by glycogen synthesis. Now here glycogen synthesis is an enzyme that will help in enlargement or elongation of chains in the glycogen synthesis. Next is formation of branches. This is also very important because we had learned in the structure that glycogen contains straight chains as well as branched chains. So here two enzymes are responsible. One is for synthesis of straight chain glycogen molecules, and also another enzyme is responsible in order to increase, or you can say in order to put the branches in this glycogen molecule. So how this has been facilitated, we will see. So this is the pathway how glycogen metabolism exactly takes place. So first glucose, obviously, because high concentrations of glucose either from the diet or already present in the body. Now this glucose is first converted into glucose 6-phosphate in presence of enzyme glucokinase. Okay, so this glucose is converted into glucose 6-phosphate in presence of enzyme glucokinase. So now the 6-phosphate molecules comes from the when ATP donates one of its phosphate molecule, right? So ATP, again it will donate its energy as well as it will donate its phosphate bond to the glucose molecules, resulting in synthesis of glucose 6 phosphate. You can say either in presence of glucokinase or hexokinase, both is okay. Okay. So next is conversion of glucose 6 phosphate into glucose 1 phosphate. You can see there is a shift in the position of phosphate groups from 6th position to 1st position. So this, now these are isomers, hence the enzyme responsible is phosphoglucomutase. The enzyme that is responsible is phosphoglucomutase. Now these glucose molecules, that is glucose 1 phosphate, whatever is synthesized, now it reacts with a UTP. So UTP, it will remove, now here also 3 phosphate, triphosphate, hence 3 phosphates are there. Among from these 3 phosphates, 2 phosphates are removed. That is, a pyrophosphate molecule is removed. This PPI is now denoted as pyrophosphate. Whenever there is elimination of 2 phosphate molecules, then whatever the phosphate, this PPI is obtained, is known as pyrophosphate, hence resulting in synthesis of UDP. Okay, so now these UDP molecules will go and replace these phosphate molecules present in the glucose. UDP molecules, whatever are obtained after removal of pyrophosphate molecules, will go and replace the phosphate molecules in the glucose. That is, from glucose 1 phosphate, the first position phosphate group is eliminated, and this UDP goes and attaches to that glucose molecule, resulting in the synthesis of UDP glucose. This facilitated in presence of enzyme UDP glucose phosphorylase. So this is the enzyme that helps in attachment of UDP to the glucose by replacing the phosphate groups. Now you can see there is a representation below UDP and a glucose. As we know, glucose is a 6-membered. So for our easy convenience in the further later steps, next steps, we represent glucose in the form of a hexagon. Okay. So now these UDP molecules in presence of this glycogen. Now whatever the glycogen I said is present, right? That is already present in the body, the fragment of that glycogen is utilized for glycogenesis. That helps in initiation of this glycogenesis. Now that fragment of the glycogen that helps in initiation of the glycogen synthesis is known as glycogenin. The fragment of glycogen present in the body that is utilized for synthesis of glycogen, that is utilized for the synthesis of glycogen is known as glycogenin. Okay. Now these glycogenin molecules in presence of glycogen initiator synthase. This is an enzyme, glycogen initiator synthase. These glycogenin molecules will interact with UDP glucose. This replacing the UDP and free glucose is obtained. So this free glucose will go and bind to the hydroxyl groups of glycogenin molecules. UDP glucose reacts with glycogenin, again replacement of UDP with this glycogenin, that is fragment, and attachment of the glucose to the hydroxyl groups. Okay, hence there are different this is glucose molecules are attached here. Now 3 UDP glucose molecules will come in contact with this glycogenin, resulting in attachment of 3 glucose molecules to the glycogenin. Hence, this glycogen primer is now synthesized. Whatever the glycogen now obtained is known as glycogen primer because it is having very less amount of glucose molecules, and these will be helps in attachment of other glucose molecules and helps in synthesis of glycogen. Now these glycogen primer molecules in presence of enzyme glycogen synthase. See, now glycogen synthase, again 13 molecules of UDP are utilized. See, 13 molecules of UDP glucose. Here the third step we had seen UDP glucose is synthesized. Similarly, in different, again the 13 molecules of glucose are utilized and 13 molecules of UDP glucose is synthesized. Now this glycogen primer attaches to these 13 molecules of UDP glucose in presence of enzyme glycogen synthase. So now this, you can see the resultant is a fragment of this glycogen only. So this is not complete glycogen. This is also again a part of the glycogen. Now the synthesis has been initiated. So you can see the numberings have been given and up to 16 because already 3 are present and again additionally 13 molecules you add. So totally 16 molecules of glucose are added to this glycogen. So now comes the part of addition of branches, right? So these branches are added up to until and unless the initial fragment of this glycogen has 8 to 10 glucose units. So you can see this is the finally got 16 glucose units. So until and unless in this fragment minimum of 8 to 10 glucose units must be present, then only this branches occurs. If these 8 to 10 units are not present, then the branches will not occur. Minimum requirement for the branches to be attached is it must contain minimum of 8 to 10 glucose units. Then only branches can be easily attached. So now already know we have seen that here now 16 molecules are present. That is much sufficient. So now the enzyme that is responsible for addition of branches is known as glucosyl 4,6 transferase. Glucosyl alpha 4,6 transferase. Now this enzyme will help in addition of branches in this molecule. Okay, so now how these molecules are added branches? You can see here already here 16 molecules are there. So out of these 16 glucose molecules only 4 of the glucose molecules, sorry, 6 of the glucose molecules are removed. 6 of the glucose molecules are removed and they are transferred at the 7th position of this glycogen chain. From the same chain only, the glucosyl residues are broken and they are transferred at the 7th position in order to form a branch. You can see there is a branching occurred between 7th and 11th glucosyl residues. So 1 to 10 is a straight chain, while from 7, 11 to 16. Now here 11 to 16 is a branched chain. So branches occur at 7th and 11th position. Now the bond formed between these two is alpha 1,6 bond. Here each and every glucose residues will be having alpha 1,4 bond, but the bond that is formed only between the 7th and 11th unit will be alpha 1,6 bond. Again from the 11 to 16 it will be 1,4 bond only. It will be no alpha 1,6 bond. Only between these two residues where the branches occur, only at that position alpha 1,6 bonds are formed. Remaining all the glucosyl molecules will have alpha 1,4 bond itself. So now this process of adding the glucose molecules in order to lengthen the chain as well as in order to initiate the branches will be continued until the final glycogen molecule is obtained. So now this is a fragment of this glycogen. So now these glycogen, these fragments go on adding the UDP glucose. They will utilize UDP glucose molecules, and these glucose molecules from this UDP glucose will be go on added on this straight chain as and also branches will be formed until and unless the required structure of this glycogen is obtained. Hence, elongation is initiated by the glycogen synthase while the branching will be initiated by glucosyl 4,6 transferase. So this is how glycogen can be synthesized from the glucose molecules. It will utilize first convert glucose into glucose 6 phosphate, then into glucose 1 phosphate. Now these 1 phosphate molecules will attach with the UDP. Will attach with the UDP, or you can say these will replace the phosphate molecules with the UDP molecules. Now these UDP glucose molecules will go and attack on the glycogenin, resulting in the synthesis of glycogen primer. Now utilizing these glycogen primer molecules, other UDP glucose molecules also come and join in until 7 to 8, minimum of 7 to 8 glucosyl residues are present. And whatever the excess of those glucosyl residues are present, that are utilized and helps in synthesis of branches in that glycogen molecule. That is facilitated with the help of enzyme glucosyl alpha 4,6 transferase. Now here you can see short fragment of glycogen is obtained having branches as well as a straight chain. Now these short fragments will continue to add on all the glucosyl molecules until the desired length of this glycogen is obtained. Hence, these enzymes, that is glycogen synthase and glycogen transferase, goes on adding and makes and helps in facilitating bond formation at alpha 1,4 position as well as alpha 1,6 position respectively, until and unless this whole glycogen molecule is obtained. So this is how glucose is synthesized. Any doubts? Understood how this glucose is synthesized? Yes? Understood? Okay. Remaining? No confusion, right? Okay. So now coming to the degradation part. Okay, so now you understood how glycogen is synthesized. Now, how this glycogen, whatever is stored in the body, how it will be degraded when body requires glucose molecules. Okay, now this degradation takes place in the liver and muscles. Specifically, it takes place in the liver and muscles, and obviously, another set of enzymes, I had already discussed this, that different set of enzymes will be responsible for degradation of glucose. The same enzymes like glycogen synthase, glucosyl 4 transferase, these all will not be utilized now. Another different enzymes will be utilized for breakdown of this glycogen into glucose molecules. So what are the different steps that are involved in now degradation pathways? So first is shortening of the chain. As we know, glycogen is a long molecule having straight chains as well as branch chains. So now all of these chains must be cut and made into short, right? So now first is shortening of the chain. Second important is removal of the branches. We had seen the branches are being specifically added using the enzyme glucosyl alpha 4,6 transferase. Now these branches must be removed such that we get only single chain of glucosyl residues, or you can say a single chain of fragment of this glycogen, such that it will help in easy degradation into individual glucose molecules. Okay, so first is we have to shorten the chain. Second is we have to remove the branches. Next is formation of glucose 6 phosphate, ultimately leading to synthesis of glucose. That is, we had seen glucose 1 phosphate is also important during the glycogen synthesis. Now, when glycogen degradation takes place, whatever the straight chain molecules of the glucose are degraded, or you can say the bonds are broken between the straight chains, results in the synthesis of glucose 1 phosphate. So now those glucose 1 phosphate molecules should be utilized for synthesis of glucose 6 phosphate, ultimately synthesis of glucose itself. Okay. Next is lysosomal degradation of glycogen. How this glycogen can be degraded in lysosomes. Okay. So we'll see. So you can see now this is a glycogen molecule. Here, for understanding purpose, we had taken only a short fragment of this glycogen molecule. Okay. So in the pink color, you can see there is a branched chain, and whatever the black glucose residues are there, those are straight chain. And the blue colored molecule is a branched glucose molecule where alpha 1,6 bond is present. Understood? These pink color is a branched chain. Whole totally it is a branched chain, while the black color is a straight chain. Now this blue and black helps in the formation of alpha 1,6 bond. Okay. So in this way you can remember. So now first there is a shortening of a chain. First step is shortening of a chain. So this takes place in presence of enzyme glycogen phosphorylase. This takes place in presence of enzyme glycogen phosphorylase. This shortening will be carried out until and unless 4 glucose molecules are present on the each side of the chain. You can see this pink colored structure as well as the black colored structure on the right hand side. So in the product whatever is obtained, there are totally 4 molecules, this blue 1 and 3. And similarly here there are 4 molecules of the black glucose residues. So this glycogen phosphorylase will help in cutting off excess of all the glucose molecules until and unless 4 residues are remained on the each side of the chain. Means here branching is also there. So we have to say each side of the chain. So until and unless 4 molecules, this is very important. 4 molecules must be remained. Until that point, this glycogen phosphorylase will act. Okay. So now second is, and whatever the glycogen that is obtained after degradation up till 4 units, that glycogen is now known as limit dextrin. So in the synthetic part, we had seen a glycogen primer was synthesized, where in case of this glycogen degradation, a limit dextrin molecule is being synthesized. Now from these limit dextrin molecules, now debranching occurs, means these branched molecules now will be removed. How these will be removed? With the help of enzyme, debranching enzyme, that is transferase. Now here the enzyme that is responsible is transferase. So what will happen? The three pink colored molecules you can see. So now the alpha 1,4 bonds are broken, and those whatever the glucose residues are present, they will be now attached to the straight chain. You can see, if you remember, in case of the synthesis also, whatever the in this step, that is after 13 UDP glucose molecules are added, now the remaining 11 to 16 molecules are removed and added to the branch. Similarly, what happens in the debranching case, these molecules will be removed and attached back to this 10th position itself. Okay, so this is the reverse way they are trying to synthesize glucose. So now whatever these glucosine residues are present in the branched chain are removed and added to the straight chain molecule, and only a single glucose unit is left out, that is having alpha 1,6 bond. Now these glucose molecules should also be removed, because now we are carrying out debranching of enzyme, sorry, debranching of the chain. So all the branches must be removed first. So this debranching is facilitated with the help of enzyme alpha 1,6 glucosidase. The enzyme that is responsible for removal of this branched glucose molecule is alpha 1,6 glucosidase. Now the role of this glucosidase molecules is to break the bond, that is alpha 1,6 bond. So this will help in synthesis of free glucose molecule. Here two important points you have to remember. Synthesis of glucose 1-phosphate and also synthesis of free glucose molecules. These free glucose molecules are obtained when the bond between the branched glucose units, that is when alpha 1,6 bond is broken, that helps in synthesis of free glucose. Directly you are getting glucose molecules. There is no synthesis of glucose 1-phosphate, no synthesis of glucose 6-phosphate also. So directly you are getting glucose residues when alpha 1,6 bonds are broken. Directly free glucose is being obtained. Now these straight chain molecules must also be degraded. Hence what happens? Here now only one alpha 1,4 bonds are present. When we try to degrade this alpha 1,4 bond in presence of enzyme phosphorylase. So in presence of enzyme phosphorylase, phosphate groups are added to each and every glucose residues. When alpha 1,4 bonds are broken and in presence of enzyme phosphorylase, all the glucosyl residues are added with the phosphate groups, resulting in the synthesis of glucose 1-phosphate. So two important points. When 1,6 bond is broken, free glucose is obtained, while when alpha 1,4 bonds are broken, glucose 1-phosphate is obtained. Now these glucose 1-phosphate undergo isomerization. This is a reversible step. Now these isomerization in presence of enzyme phosphoglucomutase, because it is a reversible step, hence results in the synthesis of glucose 6-phosphate. Now these glucose 6-phosphates can be either directly converted into glucose and sent to different organs. Now this process is being taken place in the liver. So whenever the requirement by the other organs is also there, now these glucose 6-phosphates will be converted into glucose in presence of enzyme glucose 6-phosphatase, that is specifically present in the liver itself. Whenever this glucose free glucose is now synthesized, that will be sent to different organs, majorly brain, RBCs and kidneys. These are the major enzymes that where this glucose will be sent. Or else if the requirement by these three major organs is not required, then this glucose 6-phosphate will be directed to directly glycolysis. Remember, the very first step of the glycolysis, conversion of glucose to glucose 6-phosphate. Now from glucose 6-phosphate only glycolysis will be started. No need to again utilize glucose. Understood how this glycogen degradation takes place and how this last step will help either in synthesis of glucose and transferring into different organs wherever it is required, or it will help in direct glycolysis of this glucose 6-phosphate. Okay, understood regarding glycogen metabolism? Any doubts? Any doubts or shall I continue? Are you there? Yes. Please repeat from alpha 1,6. Which step you are saying? Alpha 1,6, that is this. Can you see where I am pointing out? Yes, Afros, can you see? This step you want? Yeah. So now what happens? After debranching means whenever these glucose residues are removed and attached to the straight chain, now the remaining is only one glucose residue. Now whenever this enzyme, that is alpha 1,6 glucosidase, acts on this bond and its break, and directly free glucose is synthesized, and the left out portion is the straight chain. Now these straight chain molecules will be having glucose residues. So these straight chain molecules in presence of enzyme phosphorylase, what will happen? Phosphate molecules are added, and the bond, whatever is present, that is alpha 1,4 bond, it will be broken down and individual glucose molecules are synthesized. Now those individual glucose molecules will be added with the phosphate groups. Okay, resulting in the synthesis of glucose 1-phosphate. At the first position, phosphates are added. Now, these 1-phosphate molecules undergo isomerization, means there is a shift of phosphate from first position to sixth position. Okay, synthesis of glucose 6-phosphate results. So now these glucose 6-phosphates, either they will be converted into glucose molecules in the liver and they will be sent to different organs, or else what happens if the liver is only requirement, if the liver is in requirement of this ATP, so what will happen, these glucose 6 phosphate molecules will be diverted to glycolysis, wherein these glucose 6 phosphate molecules will be degraded to form pyruvates. From pyruvate acetyl CoA, and from acetyl CoA it undergoes TCA, resulting in oxidation, that is carbon dioxide and water. Now, whatever the coenzymes, that is NADH and FADH2, are synthesized during the whole process will be utilized for ETC and oxidative phosphorylation, resulting in ATP synthesis directly. So, whenever if liver is in requirement of energy, then these glucose 6-phosphate molecules will be directed to the glycolysis pathway. Or else, if the liver is not required ATP, then what will happen? These glucose 6-phosphate molecules will be converted into glucose and they will help in maintenance of blood sugar levels, or they will be utilized for different organs by helping the transportation of glucose from liver to the different organs. Understood? How much glucose molecules are formed? This is a fragment of this glucose, right? Now from this glycogen, you can see there are 12. Now here it is having a 12 glucose residues. Okay, so these 12 glucose residues will be forming 12 glucose molecules, including this branched chain. So totally 13. So it is just a fragment. Now this glycogen molecule is too heavy. Almost approximately it is having a weight of 300 to 400 daltons. That much is the weight of this glycogen molecule. So you can imagine how many glucose can be embedded in that glycogen molecule. So it is a polymer. So you can have n number of glucose molecules from this glycogen. Yes, any other? Fine. So shall I continue? Yes. Okay. So now coming to the regulation. Again, an important aspect regarding synthesis and degradation, how these can be regulated. Now, we had seen that different enzymes are responsible for synthesis as well as degradation. But the important enzyme that is involved in the synthesis is glycogen synthase. The very first step, that is glycogen synthase, is the enzyme that is responsible for synthesis of glycogen, and glycogen phosphorylase is the important enzyme that is involved in glycogen degradation, that is glycogenolysis. So now how these enzymes, or you can say how the mechanism of this synthesis and degradation is regulated. This is regulated by three different mechanisms. One is allosteric regulation, second is hormonal regulation, and third is influence of the calcium. So we'll see how these different Regulatory mechanisms helps in regulation of synthesis as well as degradation. So now coming to the allostatic regulation. So this allostatic regulation of the synthetic pathway, you can see the control is carried out in such a way that the glycogen synthesis is increased when the substrate availability and energy levels are high. Means when glucose is not further required by the body and excess of glucose is present, as well as ATP is sufficient in the body. Under these two conditions, means glucose concentration is high as well as ATP concentration is high. Under such conditions, the glucose now acts as a substrate molecules. So these substrates will be converted into glycogen. When the concentrations of glucose as well as ATP is high in the body, now these glucose will be converted into glycogen. So this helps in synthesis of glycogen. Now when this breakdown occurs, so this breakdown occurs when the glucose concentrations are low as well as energy concentrations are low in the body. And this takes place particularly during the fasting conditions, or also you can say during the um, nocturnal conditions. Means after having your meals at the night in the dinner, there is a long gap, right? Directly in the morning we have a breakfast, so there is a long gap, and in between this gap body, many of the organs, they are auto functioning, so they require energy. If energy stores are decreasing such conditions, now these glycogen molecules will be degraded and glucose will be synthesized, and those glucose will be utilized for synthesis of ATP, either in the fasting condition or in between the meals. So these are two conditions when this glycogen breakdown takes place. Now you can see in this image that there is a glycogen is being converted into glucose 1-phosphate. Means there is a degradation of glycogen. The conversion of glycogen into glucose 1-phosphate means there is a degradation. So degradative enzyme important is glycogen phosphorylase. Now these glycogen phosphorylase will be inhibited when glucose 6 concentration is high. Glucose 6 phosphate concentration is high, or ATP molecules are high in the high, or in the liver the free glucose concentration is high. Three conditions when glycogen degradative pathways are inhibited. One is when glucose 6 phosphate concentrations are high, when energy is high, and also when the liver concentration of a glucose, when concentration of glucose in the liver is high. Under all these three conditions, degradation of glycogen is being inhibited. And when will this glycogen inhibition increased? In the muscles, it will be increased when, means when the calcium concentrations in the muscle is high, or when AMP, that is adenosine monophosphate concentrations are high, means energy concentration is less here now. When AMP is present, that means ATP is less. Hence, in the presence of calcium as well as AMP, this glycogen degradation will be increased, and it will be decreased when ATP, that is energy and glucose concentrations are high. Okay, so this is how this glycogen phosphorylase enzymes will be regulated. So how this glycogen synthase enzyme will be regulated? You can see it, conversion of glucose 1-phosphate into glycogen. Now you are synthesizing glycogen. So how these glycogen molecules, sorry, the synthetic pathway is regulated. So this takes place in the enzyme glycogen synthase. These glycogen synthase molecules will be activated. The plus sign indicates activation with the help of glucose 6-phosphate, because high concentration of glucose is present, that is converted into glucose 6-phosphate. So now further glucose degradation is not required. ATP is sufficient, glucose is also sufficient. So not sufficient, glucose is high. So under such condition, the synthetic pathways will be activated with the help of glucose 6-phosphate, while glycogen phosphorylase, that is degradative pathways, will be activated as well as inhibited depending on the requirement of the body. Understood how this allosteric regulation takes place? Yes? Understood how this allosteric regulation takes place in the synthesis as well as degradation of glycogen? Okay. Others? Any doubts? Okay, fine. So now coming to hormonal regulation. So this is an important aspect of glucose synthesis, this glycogen metabolism. How these hormones are involved either in the synthesis or in degradation of this glycogen. So the hormones, through a series of complex reactions, bring about covalent modification like phosphorylation or dephosphorylation. You have already learned what is phosphorylation. Yes, what is phosphorylation?
Synthesis of ATP direct from the substrate.
No, that is substrate level phosphorylation. I'm talking about, yeah, that is addition of phosphate molecules. A simple definition of this phosphorylation is addition of phosphate molecules, and dephosphorylation is removal of phosphate molecules. Whatever you talked regarding synthesis of ATP directly from the substrate, that is known as substrate level phosphorylation. That is totally different. Okay, so now here phosphorylation means addition of a phosphate to the enzyme molecule and removal of a phosphate from that enzyme molecule. So how this is facilitated? So that will help in either synthesis or degradation depending on phosphorylation and dephosphorylation. Now this hormonal regulation is mediated by CAMP, that is cyclic AMP. Now this cyclic AMP has two different roles. Either it will help in synthesizing or it will help in degradation. So how? We can see in this next picture. So you can see whenever the glucagon and epinephrine levels, this both are hormones. Now these hormones, whenever the concentration of these hormones is high, so these will be released from the organs, and now these hormones will come and attach to the plasma membranes, that is on the cell. Okay, whenever these glucagon and epinephrine comes and binds to the cell molecules, in the plasma membrane, now here adenylate cyclase will be activated. Means it will be converted from inactive form into active form. Whenever glucagon and epinephrine comes and bind to the cell molecules, sorry, not molecules, the cell in the plasma membrane, now adenylate cyclase will be activated. The role of adenylate cyclase is to convert ATP into cyclic AMP. Whatever ATP is present in the cells, now those ATP molecules will be converted into cyclic AMP, that is CAMP. Role of adenylate cyclase is to convert ATP into CAMP. Now here the role of CAMP comes into account. These CAMP molecules try to activate protein kinases. CAMP molecules tries to activate protein kinase molecules. Now these protein kinase molecules causes phosphorylation. This is important. You can see there is a removal of phosphate group from ATP resulting in ADP. Now these phosphate molecules are added to glycogen synthase. Remember where it is utilized? This glycogen synthase is an enzyme that helps in synthesis of glycogen. Now CAMP, what does it does is it first activates protein kinase. Now these protein kinase molecules will cause phosphorylation of glycogen synthase molecules, sorry. They will cause phosphorylation, means they will add the phosphate groups to this glycogen synthase, resulting in inactivation of this enzyme, of the enzyme. Now these enzymes, because now this glycogen synthesis is inhibited, so glycogenesis will be inhibited, means glucose concentration will be increased rather than glycogen synthesis. Now glucose concentrations will be increased. We all are very familiar that glucagon helps in increasing the reduced blood glucose levels, right? So this is how glucagon helps. First, it will activate adenylate cyclase in the plasma membrane. These adenylate cyclase will convert ATP into cyclic AMP. Now these CAMP molecules will activate protein kinases. So these protein kinase molecules or enzymes will help in addition of phosphate groups to the glycogen synthase enzyme, resulting in inactivation of that enzyme. Hence, glycogen synthesis is inhibited. So this is how glucagon as well as this epinephrine helps in inhibition of glycogen synthesis and helps in maintenance of blood glucose levels in the body. Understood? The first role of the CAMP, how does it helps in inhibition of synthesis of glycogen and helps in maintenance of glucose levels. Understood? Regarding this hormonal regulation, a part of this hormonal regulation. Yes? Okay, now coming to how it will help in inhibition of glycogen degradation. Okay, so now similarly here again the glucagon and epinephrine. Similar process takes place. They come and bind to the cells in the plasma membrane. This adenylate cyclase is activated. Now the role of adenylate cyclase is converting of ATP into CAMP. Now these CAMP molecules again activate protein kinases. Okay, now here the different mechanism takes place. Now the protein kinase molecules, what does it do? It adds phosphate molecules to the enzyme phosphorylase kinase. That is a total different set of enzyme that is involved. Phosphorylase kinase, it is now inactive. When phosphate is added, it becomes active. So in the earlier case, we had seen that glycogen synthase enzyme, whenever phosphate is added, it becomes inactive. But in this case, that is phosphorylase kinase, whenever phosphate is added, it becomes active in nature. Now, these active phosphorylase kinase molecules helps in activation of glycogen phosphorylase. Means whatever the phosphate groups is attached to this phosphorylase kinase is transferred to glycogen phosphorylase B, and phosphate groups are added here. Now these inactive glycogen phosphorylase B is converted into active form when phosphate groups are added. So you can see two different roles of enzymes. When addition of phosphate in glycogen synthase, it will become inactive. But if same phosphate groups are added to the glycogen phosphorylase, it becomes activated. So now these glycogen phosphorylase molecules tend to degrade this glycogen. You can see whatever the glycogen is present in the liver, now we know this glycogen phosphorylase is now active. So its role will be degradation of glycogen into glucose 1-phosphate, ultimately leading to synthesis of glucose molecules. So in two different ways, these glucagon helps. Either they will inhibit synthesis of glycogen, or they will try to degrade the glycogen such that the concentrations of glucose are continuously supplied in order to meet the blood glucose concentrations. Okay, so another important point to be remembered here is, you can see this is the calcium molecules are present. Now, when this calcium, I will say how this is important. So, understood how these hormones are responsible for either inhibiting the glycogen synthesis or to carry out degradation of this glycogen such that ultimately glucose is only obtained. Yes? Any doubts? Yes, students? Any doubt? Okay, fine. Uh, Now coming to influence of calcium. Okay, so this is also important in order to maintain this glycogen metabolism, or you can say the regulate this glycogen metabolism. Now we are very familiar that the muscles. Now muscles are consisting of sarcoplasmic reticulum. These are type of the cells that are present in the muscles. Now these sarcoplasmic reticulum require calcium for their activity. Okay, whenever these calcium molecules are released from this sarcoplasmic reticulum, when muscle contracts, there is a contraction of all the cells. During this contraction, now calcium ions are released. These calcium ions bind to calmodulin. Now, this is calmodulin is one of the intermediary enzymes. So these calmodulin helps to directly activate phosphorylase kinase without the involvement of CAMP dependent protein kinase. In this you can see in this image. So first glucagon must come and attack on the cells, and then there is synthesis activation of CAMP. Now these CAMP molecules will activate CAMP dependent protein kinases. Then only there will be activation of phosphorylase kinase. But what happens in the skeletal muscles? Whenever skeletal muscles contract. So when during this contraction, calcium is being released. Now these calcium molecules will directly act on this calmodulin phosphorylase kinases enzyme. So this is an enzyme. First it will act on calmodulin. Now these calmodulin molecules will try to activate directly phosphorylase kinase. You can see they will directly add the phosphate groups into this phosphorylase kinase inactive such that it is being activated. So no role of CAMP here. Directly calcium molecules helps in activation of phosphorylase kinase. So now the role of phosphorylase kinase is to add phosphate groups to the glycogen phosphorylase B and convert it into active form such that degradation of calcium is being facilitated, glucose is synthesized. Now those glucose molecules are utilized for ATP synthesis, and those ATP molecules will be further utilized for contraction as well as relaxation of the skeletal muscles. So this is how this influence, the calcium concentrations are also important in order to carry out degradation of glycogen and resulting in the synthesis of glucose molecules. Understood? Three different mechanisms for regulation. One is hormonal regulation. Second is how allosterically these enzymes, that is glycogen synthase as well as glycogen phosphorylase, can be activated or deactivated, that is inhibited, and how the concentrations of calcium in the muscles is very important in order to degrade these glycogen molecules to glucose. Yes, understood regarding this glycogen synthesis, degradation, different pathways, different enzymes, and also the regulation of this glycogen metabolism. Fine. So this is regarding all about glycogen metabolism.